understanding RNA neurotherapeutics. RNA-based therapeutics are rapidly emerging as a class of medicines for central nervous system (CNS) disorders, leveraging the central dogma of molecular biology to modulate gene expression. RNA-based therapeutics have emerged as a transformative platform in the treatment of central nervous system (CNS) disorders, offering a level of target specificity and mechanistic versatility that surpasses conventional small molecules and biologics. By directly engaging the post-transcriptional regulation of gene expression, RNA modalities enable precise silencing, splicing correction, transcript editing, or de novo protein synthesis without the need for genomic modification. This class of therapeutics includes antisense oligonucleotides, SIOs, small interfering RNAs, messenger RNAs, microRNA, mouse modulators, long non-coding RNA, mouse modulators agents, and site-directed RNA editing systems, each tailored to address distinct molecular defects underlying neurological diseases. Their modular design and programmable sequence complementarity make them particularly suited for targeting the complex and heterogeneous gene expression landscapes characteristic of CNS pathologies. While delivery across the blood-brain barrier and immunogenicity remain key challenges, recent advancements in chemical modifications, delivery vectors, and intrathecal administration techniques have significantly expanded the clinical viability of RNA therapeutics in neurology. As a result, RNA-based interventions are rapidly advancing from experimental tools to clinically validated treatments, redefining the therapeutic potential for both genetic and acquired CNS disorders. These therapies intervene at the RNA level to either suppress, replace, correct, or augment protein expression, offering new avenues to treat diseases that have historically been intractable with small molecules or monoclonal antibodies. Recent progress in synthetic chemistry, molecular engineering, and delivery systems has catalyzed the development of RNA therapeutics that are now demonstrating clinical efficacy in neurodegenerative and genetic CNS diseases. One of the most advanced classes of RNA therapeutics are antisense oligonucleotides, APSOs, which are short, synthetic, single-stranded DNA or RNA molecules that hybridize with complementary RNA targets via Watson-Crick base pairing. Depending on their design, AS can recruit RNase H to degrade the RNA strand of a DNA-RNA duplex, modulate pre-Miri splicing by sterically blocking spliceosome components, or inhibit translation by binding to the fiber untranslation region UTR, or initiation codon. Chemically, acyls are often modified with phosphorothioate linkages and 2-O-methyl or 2-O-methoxyethyl sugar modifications to increase nuclease resistance, enhance target affinity, and reduce off-target effects. Antisense oligonucleotides represent one of the most clinically advanced classes of RNA therapeutics. They are short, synthetic, single-stranded DNA or RNA molecules that base pair with complementary RNA sequences to modulate gene expression. In the context of central nervous system CNS disorders, ASOs offer a unique ability to target previously undruggable genes through sequence-specific mechanisms, bypassing the limitations of conventional small molecules or biologics. Their mechanisms of action, chemical properties, pharmacokinetics, and delivery challenges must be precisely engineered for efficacy and safety within the CNS environment. Mechanisms of action: RNase H1-mediated cleavage, H1-mediated cleavage. ASOs designed to recruit RNase H1 acts through a degradation-based mechanism. These ASOs are typically structured as gapmers, which consist of a central DNA segment that forms the RNase H1 recognition site, flanked by chemically modified ribonucleotides that increase nuclease resistance. Upon hybridization with the target RNA, the DNA-RNA heteroduplex is recognized by endogenous RNase H1, which cleaves the RNA strand while leaving the DA intact. The ASO remains available for additional rounds of hybridization and cleavage, allowing catalytic turnover of the RNA substrate. Steric blockade of RNA processing is a mechanism by which antisense oligonucleotides, or ASOs, function without inducing RNA degradation, Instead, these ASOs physically block access to RNA binding proteins or regulatory complexes. A key example is splice-swapping ASOs, which are designed to bind to specific sequences of pre-messenger RNA, such as intronic or exonic motifs, including exonic splicing enhancers or silencers. By binding to these sites, the ASO prevents components of the spliceosome from accessing the RNA, thereby altering the inclusion or exclusion of specific exons. One clinically approved example is nusinersen, which promotes the inclusion of SN7 in the SMN2 gene, helping to restore SMN protein production in patients with spinal muscular atrophy. Similarly, translation blocking, ASOs, bind to the 5' untranslated region or to the ORG start codon of messenger RNAs. This prevents the ribosome from scanning or assembling on the RNA, effectively shutting down protein synthesis. Design considerations, particularly target accessibility, play a critical role in determining the effectiveness of antisense oligonucleotides, or ASOs. The ability of an ASO to bind its RNA target depends on how accessible the binding site is within the RNA structure. Computational tools such as mFold and RNA structure are commonly used to predict RNA secondary structures and to identify regions that are likely to remain single-stranded and exposed. These predictions are then validated using experimental methods like ShapeSeq and DMF-MapSeq, which chemically probe RNA flexibility either in vitro or in living cells. Regions that are consistently accessible are chosen to promote efficient ASO binding, while avoiding interference from complex RNA folding or competition with RNA binding proteins. Hybridization stability is a critical parameter in ASO design. Melting temperature, ETH, is optimized to fall within the range of 55 to 65 degrees Celsius to ensure that the ASO RNA duplex remains stable under physiological conditions. TM is influenced by GC content, nucleotide sequence, oligonucleotide length, and chemical modifications. Binding affinity must be high enough to ensure selective engagement with the intended RNA without significant binding to non-target transcription. Chemical modifications, particularly backbone chemistry, are essential for improving the stability and effectiveness of antisense oligonucleotides, or ASOs. One of the most widely used modifications is the phosphorothioate linkage, often abbreviated as PS. In this modification, a non-bridging oxygen atom in the phosphate group is replaced with a sulfur atom. This change significantly enhances resistance to both exonucleosate and endonucleoses, helping to protect the ASO from degradation. PS linkages also increase binding to plasma proteins, which in turn extends the ASO's half-life in the bloodstream and improves tissue uptake. However, while beneficial in many ways, this modification can also reduce the stability of the ASO RNA duplex and increase non-specific interactions with proteins. Because of this, PS content must be carefully optimized during ASO design. Chemical modifications, especially sugar modifications, play a crucial role in enhancing the performance of antisense oligonucleotides, or ASOs. Two of the most common sugar modifications are the 2'-O-methyl and 2'-O-methoxethyl groups, which are added to the ribose sugar. These changes improve the stability of ASOs by increasing resistance to nucleus degradation and raising the melting temperature, or T. In addition, these modifications help reduce unwanted immune activation, particularly through toll-like receptors 7 and 8. They are commonly placed on the flanking regions of gapmer-ASOs to protect the molecule without interfering with the activity of RNase H1. Another powerful modification involves locked nucleic acids, or LNAs. These molecules contain a methylene bridge that locks the ribose ring into a specific conformation, significantly boosting binding affinity. LNAs are often used in mixmer designs to further stabilize the ASO-RNA duplex. In specific applications, additional base modifications such as 5-methylcetosine and tricyclo-DNA are introduced to fine-tune pharmacokinetics and enhance delivery to the central nervous system. Pharmacokinetics and cellular uptake are critical factors that influence the therapeutic performance of antisense oligonucleotides, or ASOs. These molecules enter cells primarily through various endocytic pathways. Uptake can occur via absorptive endocytosis, scavenger receptor mediated internalization. Among these, scavenger receptors such as SRA1 and SRB1 play a key role in mediating the efficient uptake of phosphorothioate modified ASOs. Once internalized, ASOs are generally trapped within endosomes, and only a small portion escapes into the xylosol or nucleus. This limited escape reduces the overall bioavailability at the intended RNA target. In terms of tissue distribution and half-life, phosphorothioate ASOs exhibit strong binding to plasma proteins like albumin. This interaction reduces renal clearance and promotes broader tissue distribution, including delivery to the central nervous system. For targeting the CNS, intrathecal administration is typically used. This method involves direct infusion into the cerebrospinal fluid, allowing the ASO to bypass the blood-brain barrier. ASOs show prolonged half-lives in the CNS, often persisting for weeks to months. This extended duration supports infrequent dosing schedules and enables long-term modulation of gene expression. Immunogenicity and off-target effects are important considerations in the development of antisense oligonucleotides, or ASRs. Unmodified or poorly designed ASOs can activate the immune system by interacting with pattern recognition receptors. In endosomes, Toll-like receptors 3, 7, and 8 recognize specific RNA sequences and trigger immune responses. In the cytoplasm, receptors such as RIG-I and MDA5 detect double-stranded or highly structured RNA, leading to the activation of interferon-stimulated genes and the release of inflammatory cytokines. To reduce these immune reactions and improve tolerability, chemical modifications are applied to the ASOs. Off-target effects may occur when ASOs partially match unintended RNA transcripts or interact non-specifically with RNA-binding proteins, including those found in nucleus structures like paraspeckles. These risks are carefully managed using computational transcriptome-wide screening and experimental biochemical assays to ensure specificity and safety. CNS-specific optimization is essential for the therapeutic use of antisense oligonucleotides, or ASOs, in neurological disorders. These molecules must be chemically stable in the cerebrospinal fluid, show low immunogenicity in both glial and neuronal cells, and effectively distribute throughout the central nervous system. Delivery is typically achieved through intrathecal or intraoral ventricular injection, which allows direct access to the CNS. In some cases, ASOs are taken up by peripheral axon terminals and transported retrogradely to central neurons. This transport mechanism is especially important in motor neuron diseases like spinal muscular atrophy. Developing ASOs for CNS applications requires an integrated approach that combines RNA biology, nucleic acid chemistry, pharmacology, and neuroanatomy to ensure both safety and efficacy. In addition to ASOs, small interfering RNAs, or siRNAs, represent another powerful class of RNA therapeutics. These are short, double-stranded RNA molecules, typically 21 to 23 nucleotides in length. Inside the cell, siRNAs are processed so that the antisense strand is loaded into a protein complex called RISC-ARC, the RNA-induced silencing complex. Once loaded, the siRNA guides RISC to a complementary messenger RNA, where the enzyme argano2-2, also known as AGO2, cleaves the target transcripted. This process leads to precise and potent gene silencing at the post-transcriptional level. Although siRNAs offer high sequence specificity, they are vulnerable to degradation by nucleases. To overcome this, they are often formulated with lipid nanoparticles or chemically conjugated to cell-penetrating ligands for protection and enhanced delivery. Messenger RNA, or mRNA, therapeutics represent a fundamentally different approach compared to anti-sensing gene expression RNA interference strategies. Instead of silencing gene expression, mRNA therapies work by introducing synthetic messenger RNAs into cells to produce a therapeutic protein directly at the site of action. These engineered mRNAs include optimized features such as modified cap structures, including anti-reverse cap analogs, as well as fine-tuned 5' and 3' untranslated regions and polyad tails. Together, these elements improve both the stability of the mRNA in the cytoplasm and its translation efficiency. To avoid triggering immune responses, mRNA molecules are often modified with nucleosides like pseudouridine and N1-methylpseudouridine. These changes help reduce recognition by innate immune sensors, including toll-like receptors and cytoplasmic detectors like RIGI. In the central nervous system, RNA therapeutics are currently under investigation for a range of applications, such as enzyme replacement, delivery of neurotrophic factors, and even as vaccines for treating brain tumors. Messenger RNA therapeutics for CNS applications. Introduction. Messenger RNA, or mRNA therapeutics, represent a unique class of RNA-based medicines. Unlike other RNA modalities that silence gene expression, RNA therapies introduce synthetic messenger RNA into cells to produce therapeutic proteins directly at the site of action. In the central nervous system, this strategy is especially promising for treating disorders caused by missing or dysfunctional proteins, for delivering neuroprotective factors, or for developing targeted immunotherapies such as cancer vaccines. One of the key advantages of mRNA therapeutics is that they enable protein expression without integrating into the genome. This feature offers a strong safety profile and makes the technology adaptable to a wide range of diseases. However, developing mRNA therapies for use in the brain and spinal cord presents several challenges, including maintaining RNA stability, minimizing immune activation, and achieving effective delivery to the target tissue. RNA structure and engineering. Therapeutic mRNA molecules are carefully designed to maximize stability, translation efficiency, and immune tolerance. The structure of a typical mRNA includes several important components: a 5' cap, a 5' untranslated region, or UTR, a coding sequence, a 3' UTR, and a poly-A tail. The 5' cap plays a vital role in protecting the mRNA from degradation and ensuring it is properly recognized by the ribosome. To improve this function, modified cap structures such as anti-reverse cap analogs, or ARC-RRAs, are often used during in vitro transcription. These help ensure the cap is in the correct orientation and improve binding to the translation initiation factor known as EIF4. In some cases, newer proprietary capping methods like CleanCap are used to further enhance protein production and reduce detection by immune sensors. The untranslated regions flanking the coding sequence are also critical for performance. The 5' UTR is optimized to minimize RNA folding, which makes it easier for the ribosome to scan the sequence and begin translation. The 3' UTR is engineered to include elements that regulate how long the mRNA lasts inside the cell and where it localizes. These may include all-rich elements or specific microRNA binding sites. The coding sequence itself is codon optimized to match the preferred usage of transfer RNAs in the host species. This reduces the chances of ribosomal stalling and improves overall translation. Finally, the poly-A tail, usually between 100 and 120 adenine bases, protects the mRNA from degradation at the 3' end and helps form a looped structure by interacting with poly-A binding proteins. This circularization boosts the efficiency of protein production, nucleoside modifications, and immune evasion. Unmodified synthetic messenger RNA is naturally immunogenic. When introduced into the body, exogenous RNA can be detected by innate immune receptors. These include toll-like receptors, specifically TLR3, TLR7, and TLR8, located in endosomes, as well as cytoplasmic sensors such as RIGI and NDA5. Activation of these receptors leads to the production of type I interferons and pro-inflammatory cytokines. This immune response not only reduces mRNA translation, but also promotes rapid degradation of the transcript. To address this, mRNA therapeutics incorporate chemically modified nucleosides that mimic natural RNA modifications found in human cells. The most commonly used are pseudouridine and N1-methylpseudouridine. These modifications reduce the rigidity of base pairing and alter RNA secondary structure in ways that make it less recognizable to immune sensors. without interfering with the ribosome's ability to initiate translation. In addition to reducing immunogenicity, these modified nucleosides enhance MRM stability by limiting the activation of RNA-degrading enzymes and suppressing the recruitment of RNA decay complexes. During in vitro transcription, these modifications are built into the RNA by incorporating modified nucleotide triphosphates. To further purify the final product, chromatographic techniques such as reverse phase high-performance liquid chromatography, or RPHPLC, are used to remove double-stranded RNA contaminants that can arise during transcription. This extra purification step improves the safety and translation efficiency of the mRNA product and protein expression in CNS cells. Once therapeutic mRNA is delivered to the cytoplasm, it bypasses the nucleus entirely and directly engages with the host's translation machinery. Translation begins with the formation of the 43S pre-initiation complex, which includes the 40S ribosomal subunit. Initiation factors such as EIF2 bound to GTP and initiate a methionine tRNA, along with additional scaffold proteins. This complex binds to the mRNA's 5' cap and scans the sequence until it encounters an ORG start codon in a favorable COSIC consents context. From there, the process continues through elongation, mediated by elongation factors EEF1 and EEF2, and concludes when the ribosome reaches a stop codon. The newly synthesized protein is then released and properly folded with the help of molecular chaperones. In central nervous system applications, the specificity of protein expression is dictated by the delivery vector and the route of administration, rather than the mRNA sequence itself. A range of CNS cell types, including astrocytes, neurons, oligodendrocytes, and microglia, are capable of translating exogenous mRNA. However, the efficiency of translation can vary depending on cell-specific uptake mechanisms and internal conditions within the cytoplasm. Importantly, mRNA therapeutics are transient by nature. This temporary expression is actually an advantage in the CNS, where long-term or uncontrolled protein production can lead to side effects such as excitotoxicity, inflammation, or unwanted neural remodeling. Typically, protein expression lasts from several hours to a few days, depending on the mRNA design and the cell's internal environment. Applications in CNS disease. Messenger RNA therapeutics are being explored across a wide range of central nervous system disorders. In enzyme deficiency diseases such as lysosomal storage disorders, mRNA can be used to encode the missing enzyme and restore metabolic function directly in the CNS. For example, preclinical studies have tested mRNA encoding BGL choronidatus and metachoronic leukodystrophy, respectively. Beyond enzyme replacement, mRNA constructs have been used to deliver neurotrophic factors, proteins that support neuron survival and repair. These include brain-directed neurotrophic factor, or BDNF, glial cell line derived factor, or GNFS, and insulin-like growth factor 1, or IGF-1. Such therapies are under investigation for conditions like Parkinson's disease, amyotrophic lateral sclerosis, and spinal cord injury. RNA-based immunotherapies are also showing promise for neuro-oncological applications. These therapies act as vaccines, encoding tumor-specific neoantigens or shared cancer proteins. The goal is to trigger a strong T-cell response against malignant brain cells, such as those found in glioblastoma. Depending on the tumor's accessibility to immune cells, these vaccines can be delivered either locally to the CNS or systemically through peripheral administration. Delivery considerations. Delivering messenger RNA to the central nervous system presents significant challenges. This is due to the size and negative charge of the mRNA molecule, as well as its vulnerability to degradation by nucleases. To address these obstacles, mRNA is typically encapsulated in specialized delivery systems, most commonly lipid nanoparticles, or LNPs. These structures protect the mRNA from enzymatic breakdown and help it escape from endosomes once inside the cell. LNPs are composed of several key components: ionizable lipids that bind and condense the mRNA, helper phospholipids that support vesicle structure, collateral for membrane stability, and polyethylene glycoconjugated lipids that improve circulation time and prevent aggregation. Together, these ingredients self-assemble, into spherical vesicles capable of fusing with cellular membranes to deliver their polydor. To improve delivery specifically to the brain, LNP surfaces can be modified with targeting ligands. Examples include transferrin and peptides derived from the rabies virus glycoprotein. These ligands help the nanoparticles cross the blood-brain barrier by promoting uptake through receptors on brain endothelial cells and neurons. An alternative strategy is direct delivery to the central nervous system. This is achieved through intrathecal or intracerebroventricular injection, which bypasses the blood-brain barrier entirely. These routes provide immediate access to the cerebrospinal fluid and the surrounding brain tissue, greatly enhancing the efficiency of CNS. Messenger RNA, mRNA, therapeutics offer great promise for treating central nervous system CNS disorders by enabling direct, transient protein expression within the brain and spinal cord. Cellular uptake of mRNA occurs primarily via endocytosis, but efficient endosomal escape remains a major limiting factor for cytoplasmic delivery. Ionizable lipids in lipid nanoparticles become protonated in the acidic endo-CS virus environment, destabilizing membranes and releasing mRNA into the cytosol. Alternative delivery platforms, including exosomes, cell-penetrating peptides, and polymeric nanoparticles, are also being developed, each with distinct properties affecting biodistribution, half-type, and cell-type targeting. Advances in mRNA engineering, chemical modifications for immune evasion, and delivery technologies have transformed mRNA from a fragile immunogenic molecule into a potent therapeutic agent. Active research continues in areas such as enzyme replacement, neurotrophic factor delivery, and mRNA-based immunotherapies for CNS diseases. Realizing the full potential of mRNA in neurology will depend on the integration of molecular neuroscience, RNA chemistry, and targeted delivery science. Other RNA-based approaches. Other RNA strategies include using microRNA mimics and inhibitors, as well as long non-coding RNA modulators. MicroRNAs, or miRNAs, are short RNA, about 22 bases long, that regulate gene activity by binding to matching sequences in messenger RNAs. This usually happens at the end of the mRNA and leads to the blocking or breakdown of the message. To stop specific mRNAs from working, scientists use special blockers called anti-miRs. These are often made stronger and more stable with chemical changes like locked nucleic acids. On the other hand, when miRNAs are too low in a disease, synthetic mimics can be used to bring their function back. Long non-coding RNAs, or link/RNAs, are much longer, over 200 bases, and they control gene activity in different ways. These include changing how DNA is packaged or how messages are processed. Introduction to non-coding RNAs in CNS diseases. Besides messenger RNA and antisense drugs, small and long non-coding RNAs are now seen as powerful tools for controlling genes. These RNAs don't make proteins, but they control many steps in how genes are turned on and off. In the brain and nervous system, where gene activity changes over time and across cell types, changes in these RNAs have been linked to diseases. These include Alzheimer's, autism, brain tumors, and injury. New therapies are being developed that target these RNAs with increasing precision. The field is growing quickly. MicroRNAs. How they work. MicroRNAs are tiny RNA molecules, usually 20 to 23 letters long. They stop certain proteins from being made by binding to messenger RNAs and preventing their use. MicroRNAs start as long transcripts called primary miRNAs. These are cut in the cell nucleus by proteins called DROCHA and DGCR8. They are then sent out of the nucleus and cut again by a protein called DICER. This makes a double-stranded RNA. One strand is kept and loaded into a complex called RISC. RISC then looks for messenger RNAs with matching sequences and stops them from being used or helps break them down. blocking overactive myosinase with anti-myo. When a microRNA becomes too active in the brain, it can block important genes. To fix this, scientists use anti-myors, short pieces of RNA that bind to the microRNA and stop it from working. These anti-myors are often chemically changed to make them stronger and last longer in the body. For brain use, they can be linked to targeting molecules or packed into special particles. These help them get across the blood-brain barrier. In animal models, anti-myors have been shown to reverse disease effects in conditions like epilepsy and brain cancer, replacing lost myrinase with mimics. In some brain diseases, helpful myrinase are lost. Synthetic mimics can bring them back. These mimics look like natural myrinase, but are chemically stabilized. Only the working strand is designed to be used. The other strand is broken down. Delivery into the brain is a big challenge, so scientists use special injections, viruses, or peptides to get them to the right place. Promising results have been seen in animal models using mimics for stroke, memory, and nerve protection. Long non-coding RNAs in the brain. LancRNAs are longer RNAs that control gene activity in many ways. Unlike microRNAs, which mainly act after a message is made, our LancRNAs can control gene activity at every stage. They can affect how tightly DNA is packed, how genes are read, how messages are cut, or how stable the messages are. Their actions depend on their structure and how they interact with other molecules. In the brain, LancRNAs help shape neurons, support cells, and influence inflammation. Each LancRNA can have several effects depending on where and when it is active. Using antisense to knock down long non-coding RNAs. Some long non-coding RNAs are too active in brain diseases. Scientists use antisense oligonucleotides, or ASOs, to silence them. These are short pieces of RNA that bind to the long non-coding RNAs and lead to its breakdown or block it from working. Because many long non-coding RNAs stay in the cell nucleus, ASOs must be designed to reach that area. This can be done with certain chemical changes. In Alzheimer's models, ASOs targeting the long non-coding RNAs, base 1, BACs, have reduced harmful protein buildup. CRISPR and RNA editing for long non-coding RNAs. New tools like CRISPR are being used to target long non-coding RNAs without changing DNA. A special version of CRISPR called dCas13 can bind to RNAs and block or modify them. Another method uses enzymes like ADAR to change specific letters in the RNA message. These methods can be turned on or off and directed to specific cells. This is important in brain diseases like tumors or inflammation. These tools offer a more flexible and reversible approach than editing DNA. Blocking RNA-protein interactions. Long non-coding RNAs often work by interacting with proteins. Blocking these interactions can stop the long non-coding RNAs from doing its job. This can be done with small molecules or special oligos. First, scientists need to map where the RNA and protein touch. This is done using methods like clip-sequid. In brain diseases, this strategy may stop harmful long non-coding RNAs from working with key regulators of inflammation and gene silencing. RNA editing therapeutics in neurological disorders. RNA editing is a powerful way to change RNA messages after they are made, without altering the DNA. The most studied type is called A to I editing, where an enzyme changes adenosine to inosine in RNA. The inosine is read as guanosine by the cell, which changes the protein that is made. This lets scientists correct single-letter mistakes in RNA, offering a safer option than permanent DNA editing. In the brain, where RNA diversity is critical for neurons to work properly, RNA editing is being tested for many diseases. These include neurodegenerative, how ADAR enzymes work. The enzymes that do A to I editing are called ADARs. Humans have three kinds: ADAR1, ADAR2, and ADAR3. Only ADR1 and ADAR2 actually perform editing. ADAR3 seems to block editing, and is mostly found in the brain. ADARs work by finding double-stranded parts of RNA and flipping specific adenosines into inosines. ADAR2 is the key editor in the brain and is important for editing a receptor called GluA2, editing the GluA2Q/R site. In the GluA2 RNA, there is a site called Q/R that needs to be edited. Editing changes a glutamine codon into an arginine codon. This single change makes the AMPA receptor safer by stopping calcium from flooding into the neuron. Without editing, too much calcium enters and damages brain cells. This can happen in diseases like ALS, Alzheimer's, and after stroke. Restoring editing at this site can protect neurons in animal models. Therapeutic RNA editing platforms. There are two main ways to use RNA editing in therapy. The first uses special guide RNAs to attract the body's own ADAR enzymes to the target RNA. These guides base pair with the target to form a double-stranded structure. They are designed to place the target adenosine in the right position for editing. The second method brings in custom-built ADAG proteins. These are fused to RNA targeting tools like Cas13 or peptides that help guide them to the right spot, ensuring precision and safety. For RNA editing to work well, guide RNAs must form stable but specific pairs with the target. Scientists often include loops or mismatches in the guide to make it selective. A big concern is off-target editing, changing the wrong adenosines in other RNAs. This can lead to bad protein changes, faulty splicing, or disrupted regulation. To reduce this, guide RNAs can be shortened or chemically tweaked. RNA sequencing is used to check the whole transcriptome for unintended edits. Editing in other brain targets. Besides GluA2, other brain RNAs can be edited for therapy. One example is the 5-HEC-TOL2C receptor, which affects how brain cells signal. Editing changes how this receptor interacts with other proteins. Other targets include ion channels, scaffolding proteins, and disease-linked genes. Editing could help fix point mutations in rare diseases like Rett syndrome, Dravet syndrome, and some epilepsies. This makes RNA editing a powerful tool for inherited brain disorders. Delivering RNA editing tools to the brain. Getting RNA editors into the brain is difficult because of the blood-brain barrier. For guides that use the body's own ADARs, scientists use lipid nanoparticles, viruses, or synthetic oligos. AAV vectors are popular because they work well in brain cells. Certain types like AAV9 and AAV-PHB can reach wide areas of the CNS when injected into the bloodstream or spinal fluid. For engineered ADAR systems, AAVs can carry the editing enzymes and guides together. This has worked in animal models to correct editing in specific brain regions, making editing safer and more precise. It's important that the editing system is active only in the right place and at the right time. To do this, scientists use brain-specific promoters and on-off switches. Local injections into spinal fluid or brain tissue also help focus the treatment. Getting the dose and timing right is key to avoiding side effects, the risks and reversibility of RNA editing. Even though RNA editing doesn't change DNA, there are risks. Editing the wrong site could produce faulty proteins or disrupt gene regulation. Also, the double-stranded RNA formed during editing might trigger the immune system. This could block translation or cause inflammation. To avoid this, researchers are developing more accurate ADARs and carefully designed guides. The good news is that RNA editing is reversible. Edited messages are eventually broken down and the effect fades. Clinical RNA drugs in CNS diseases. Some RNA-based drugs are already in use for brain and nerve diseases. One example is nusinersen, which helps kids with spinal muscular atrophy. It changes how a gene called SMN2 is spliced to produce more of a needed protein. In Huntington's disease, antisense drugs target the faulty Huntington gene to reduce toxic protein buildup. In ALS, drugs aim at mRNAs for SOD1 TDP43, and FUS. Some companies use viruses to deliver RNA therapy directly into the brain. Challenges in CNS delivery. The blood-brain barrier makes it hard to get RNA drugs into the brain. Lipid nanoparticles can be coated with special molecules to help them cross. Another approach is to use viruses like AAV9 or AAVrh10, which are good at reaching brain tissue. In some cases, doctors inject drugs directly into the spinal fluid or brain. Other new carriers include exosomes, tiny natural vesicles that can carry RNA and cross into the brain. These are being engineered for better targeting and safety. Future of RNA drugs for the brain. RNA drugs offer unmatched control over gene activity. They can silence, fix, replace, or edit RNA messages inside brain cells. Unlike pills or protein drugs, RNA therapeutics can reach targets inside the cell, or even the nucleus. They are especially useful for diseases with known genetic errors. RNA therapies are flexible and easy to redesign by changing the sequence. This makes them ideal for rare diseases and personal treatment. Most RNA drugs don't change DNA, so they're considered safer for long-term use. New types of RNA-based treatments are being developed for many brain conditions. Better delivery and design tools. New ways to deliver RNA drugs are helping solve the problem of reaching the brain. These include safer lipids, smart viruses, and chemical tricks that improve targeting. Better computer tools are also making it easier to predict RNA structure and avoid off-targeted effects. These improvements are speeding up the development of new therapies. In the future, RNA will be at the center of many treatments for brain diseases. It will let doctors control gene expression with great precision and fewer side effects.